First, I'd like to thank our sponsor, Hewlett-Packard Enterprise, for sponsoring this session. Second, please put your questions into the chat box. We will, get to them as time allows. We now have our next speaker, Dr. Fritz Sedlazeck from the Human Genome Center here at Baylor College of Medicine. He will be talking about today success and remaining challenges of SARS-CoV-2 testing, research, and communication. Fritz, thank you for being here today. Hi, Fritz. We can't hear you. You're on mute. Fritz?
Yep. Sorry about that.
No, don't worry. Just start all over.
Yep. No problem. Sorry about that. So, welcome everyone. Thanks again for giving me the opportunity to present my work. As you can see, we're looking for a postdoc to join our group. So if you're interested, please ping me. So over the last 8 or 9 months, as you know, we are sadly in this kind of global pandemic scenario of COVID-19, where we have worldwide around 43 million cases and sadly 1.2 million deaths on the whole world. But so far, what have we learned? What have we gained knowledge about this COVID virus? And I'm trying to kind of give a broad introduction here. So what do we know about the SARS-CoV-2 virus, which is actually standing for Severe Acute Respiratory Syndrome Coronavirus 2? Well, it's a 30 kb long single-stranded RNA virus that makes it also one of the largest RNA viruses that we know of. The schematic here on the left side just shows you roughly the structure, where we have the RNA in the middle, surrounded by a hull, having different proteins that have different functions, for example, spike protein, and so forth, for docking and infecting the cells. What is really fascinating, actually, is the structure of this RNA genome, where we have this ORF1 and ORF2 that are actually getting directly translated in the genome. But maybe even more so is the complex and interesting structure of how it transcribes and translates the hull proteins, which are the spike proteins here in green, and other components. And it's interesting that here on the left you see a TRS side. This gets always coupled together with the part of, for example, the spike protein, and gets translated. And that kind of forms interesting structures. And there's a whole nother lecture to be learned about the efficiency of viruses in the regulatory mechanism of all these different genes. So 93% of all the transcriptions are really just the spike protein and the 3A compound here, which is bound together with the TRS. What do we know about the life cycle? Well, as the previous talk already said, like ACE2 is really important throughout the whole life cycle of SARS-CoV-2. It binds on the ACE2 receptor, and just as an interesting side fact, ACE2 is often found in the lung and in the gut, and as you follow the news, that's really most of the impact of SARS-CoV-2 that is recognized. It then starts the translation of ORF1A and 1B that is later needed, and then synthesis of the nested mRNAs. These are the spike proteins and other compounds to form the hulls. This is then getting translated and repacked through the Golgi apparatus, and then forming these vesicles to then be released and infect other cells in our body. So the virus has basically 2 objectives. First is to efficiently utilize the host machinery, which is in this case sadly our cells, and then also downregulate and escape our host immune response, which SARS-CoV-2 is apparently very effective in. What do we know about the origin of this virus? When we compare the SARS-CoV-2 genomes to other related genomes, for example, the MERS-CoV, here we just see 50% similarity. The SARS-CoV-1 virus, that's 80% similarity. But then really when we look at one of the bad COVID viruses, this is similar, 96% identity between that and the SARS-CoV-2 viruses, and more reports are mounting that actually there was a transition from the bad virus into Bengalis, and for the virus similarity itself is a little bit reduced. What is interesting, that's where the hypothesis I think is coming from, is that the interface sites are basically highly similar to 99%. So over the past month, what has been mounted is really kind of a global effort in terms of research and working together. People all over the world submitted de novo assembled genomes from SARS-CoV-2 to TriSeed on NextStrain is a nice interface where you can look at this all data sets. So almost per hour, we're getting new strains assembled and deposited in this database throughout the whole world. They have multiple interactive tools looking at the phylogeny of these viruses and also of the diversity. And I think right now we have around 8,000 or 9,000 strains, maybe even more than that already. So what I want to look into today with you guys is the following challenges that we were faced with beginning of February, March, which were obvious, like how can we scale testing to rapidly diagnose people if they carry the SARS-CoV-2 or not. And I think this is really essential because, as you know, from the point of infection to the point of that you show symptoms, that can really be 5 to 10 days, depending on the severity of the infection. And so for 5 to 10 days, you can infect other people, but you don't maybe even feel the symptoms. Then we also looked into what happens when we actually infected, giving our transcriptions and gene regulation. Then how does this virus evolve, kind of how diverse is this virus, and then I'm going to have one slide a little bit about future research that we're going to. So the first thing is really how can we scale and how can we test? And here the Human Genome Sequencing Center that I'm a part of really kind of took a kind of good step forward. So in March, the CMR, which is another department of Baylor College of Medicine, and also really launched the COVID-19 testing, where we developed qPCR pipelines for rapid testing. We got FDA pre-approval in April, and then in May, we actually launched the first contract with the city of Houston to provide SARS-CoV-2 testing. And up to September, I have to update the slide since we are now in October, but until September, we tested so far 60,000 tests or 60,000 cases and had around 10,000 positives. So these cases are mainly coming in from mobile testing sites around the city. And 10,000 positives are stored for further research for testing and investigating diversity of the virus. And here the HGC, I think, really kind of made a huge effort, and I cannot kind of underline how kind of proud I am of all my colleagues here. So we have really a timely return of results. We can get the samples and report the positivity to patients within 24 to 48 hours, which is not kind of an easy task. What we did, we built an end-to-end informatics reporting platform to enable this. So it's from the sample intake over the qPCR testing with robot equipment, and then like a whole LIMS system together with a sample report. I'll just give here one example of a negative result reported back to an individual. And this kind of large scale of this workflow, as well as our previous experience, made us now one of the contracts, or I think the only contractor right now with Harris County in the city of Houston that we are particularly proud and thankful of. And of course, we have state-regulated reporting included. So, but like as a scientist, I'm more and more interested in like what actually happens once we get infected. How does the whole system react to this? And for that, I teamed up in early February, March. I teamed up with a team from Cornell that had funding to sequence already from private investors that sequenced already 735 potential COVID cases and controls. So these were people in New York City that felt symptoms and walked into the clinic and wanted to get tested. We tested them with qPCR as well as with another assay, which is a LAMP assay, and then we're able to sequence the entire nasal swab. And what is so particularly interesting on this thing is that currently researchers and many universities are kind of only restricted to sequence the virus component of these nasal swabs. But in this case, we had the exception and the allowance to sequence not just the virus, the SARS-CoV-2 virus, but also the human component, as well as other components of this nasal swab with Illumina RNA sequencing. So you can see here a kind of a quick screen. On the x-axis, you would have the different samples. On the y-axis, you have the concentrations of bacteria, humans, and then we clustered that in qT-PCR negative and qT-PCR positive. Overall, we identified 9 samples here in orange that were qRT-PCR negative, but also showed signs of SARS-CoV-2. So since this is RNA-seq data, we can identify the expression of the genes and cluster them accordingly to severity of the viral load. So here I'm just showing again the ACE2 receptor, again just as a reminder, this is really an important gene and receptor throughout the life cycle of SARS-CoV-2. We had an enrichment of that in the patients with a higher load of viruses, which kind of makes sense. Just as kind of some caveats or snapshots, we also looked at IFI6 and IFI27, which both play a role in cell alloptosis, so these slices, which are also increased in the severity of the viral concentration, which also makes sense. And then here, the last gene I'm going to show is HER6, which is upregulated as a immune response gene, which again we could see kind of clearly from our data. On the downregulated side, we had oligofactory receptor pathway, which was downregulated, which kind of correlates to the reports that are mounting that people are losing a sense of smell. We had TMPRSS11B, which is a cell growth gene pathway, and also LS2, which is responsible for developing red blood cells, which are all downregulated. If you're interested, the group from Cornell is hosting this data for you to explore. If you have a particular gene that you're interested in, for example, I looked it up, the HLA-F gene, which is an important gene for immune response, you can produce these plots on your own. So given the patient data and given the RNA-seq data sets, what did we learn? Well, we saw a high increase of risk effects on males and people that had diabetes. You can imagine these early collected data sets were not very rich in meta information, so that was a really big struggle for us. And I hope this kind of improves in the future. And then on risk of mortality, obviously age has been reported throughout the media. We see still an enrichment over the males as well as diabetes and overweight cases. So this is all on the human side, what is happening over the infection, but like how does this virus actually evolve and how does it kind of adapt to different scenarios? So as again, like from these samples, we also had the viral component sequence, so SARS-CoV-2. I'm not going into different bacteria or other elements that we found in these samples, but just in the SARS-CoV-2, we were able to fully assemble the length, full-length assemble this genome for 155 cases, even the 9 cases that were RT-PCR negative. And then we had 97 samples where we weren't able to fully assemble them, and that was mainly due to uneven coverage or lacking of some spots. Therefore, they were not fully assembled. Altogether, when we built the phylogeny and compared our samples here on the left to the samples from Chasiad back in March this year, we could identify several different interesting points. First of all, the majority of our samples, so 123 out of the 141, were clearly identifiable as the A2 clade, which is originating from Western European. This nowadays fits nicely with what we know, that the virus was mainly coming in from Europe, actually into New York. However, we also identified a few strains which carried the L strain, which were also correlated to severe cases in Wuhan. That was particularly interesting. But like the majority of the strains were really coming from Europe, given our analysis. What did we learn further? Well, actually, when we looked more closely, we identified around 6 significant enriched single nucleotide variants across the samples, highlighted here in red, that are all in the A2 subclade. So we think due to the 6 enriched clades, this is kind of a new subclade of the A2 because it was so consistent across different samples. And it's interesting that actually this 6 mutations compared to the A2 subclade occurs in 84% of all GIAD New York City cases, and also in 92% of all the A2 New York City cases that are reported from CNA and New York University. For me, particularly interesting was also the identification of structure variants and single nucleotide variants in the respect of lower frequency. We call it variant allele frequency, which represents basically how many reads per sample supports a certain difference. And this was particularly interesting because on a typical virus, you wouldn't expect to identify such an amount of low frequency or low variant allele frequency mutations, which kind of highlights the potential to either a high mutation rate or a co-infection of multiple virus SARS-CoV-2 subclades or multiple SARS-CoV-2 viruses on the same host. And we also identified significant enrichment of this heterozygous variants in the qPCR negative cases that we had just 9 from, but it still turned out to be significant. So when we looked at this, I give you here like 3 samples where you have on the right side the highest variant allele frequency of 1. So every read would agree on that variant. And then on the left side, and then colored in red, are those variants that I'm currently talking about. We had around 25% variant allele frequency of these single nucleotide variants, which was very interesting to observe. And I think by unexpected, I think there are now like coming out 2 or 3 other papers that are also reporting this observation. Together with Todd Triangen's lab at Rice University, we further studied this diversity across 151 samples. So here we took the 140 samples from New York City together with 11 samples that we had finally sequenced at that time here in Houston. That was around March. To identify further single nucleotide variants and structure variants. So structure variants are larger mutations of the genome of multiple base pairs, and actually most of the interest of my group, where they have shown particular key impacts on multiple organisms. So that's why we were interested to study these 2, and including low frequency. And we also looked into how this might or might not impact our RT-qPCR. And I think Nick yesterday gave a talk, a nice talk about this already. So I'm just going to browse through it. So we looked at the interhost diversity. What was noticeable was that we had multiple. So first of all, what was notable that the Houston and the New York City SARS-CoV-2 data sets were hugely similar. Just note that the Houston one was just around 10 or 11. The New York City one was 140. We saw a huge difference, though, in the allele frequency variance in the green one, which was SARS-CoV-1, whereas MERS had a similar level of low frequency mutations. A full disclaimer, for SARS-CoV-1 and MERS, there were not many public available data sets available for that study, sadly. When we look at the transitions, we see most of the time a C to T transition as well as a T to C transition, which is kind of similar between Houston and New York City, especially on the consensus level. However, on the low frequency level, that changes a little bit, but not dramatically. And then we also looked at paired mutations, which was very particularly interesting. So we identified multiple 230 single nucleotide variants that were initially low frequency at our case, but we found them in GCAT, which highlights that there were consensus differences in the database. We also found 2 single nucleotide variants that had different variant allele frequencies across the different patients, where we took the nasal swabs from, which was kind of very curious to see how in some patients this particular variant was very lowly abundant, and then had a much higher abundance of fixation in other patients. And using this data, we identified potential pairs of transmissions between different persons. However, for a more detailed study and more comprehensive study, we would need more, again, more meta information that I think is now available. So lastly, what my group particularly was interested in looked at are the structural variants, as I said before. And this is kind of immense tricky here because, as I said in the introduction, we have this kind of strange pattern of transcription since it's an RNA virus, where we have a start site here in the beginning of the virus that is linked to the different proteins or different genes of the virus in the end of the molecule. And this kind of transcription is the case in 92%. And this forms, of course, also nicely a structural variance signal. So we had to decipher between structural variance and actual transcription signals. So we filtered the structural variance site. We found a total of 247 deletions and 23 insertions that we found trustworthy. We identified a total of 8 deletions that were actually with an allele frequency of 20% or higher. As you can see, prevalent and spike. When we normalize this by the length of these genes, we see NSP11 and NSP12 is enriched for these mutation events, giving the length compared to the other genes. So bring it to an end. What are the lessons learned? Well, higher diversity. I think the diversity is higher in the viruses than expected. We learned a lot, but of course, many open questions and challenges are remaining. There we have continuously new recordings and new details about SARS-CoV-2, about the diversity about the infection. And what I really learned over the last 8 to 9 months, and that's I'm particularly thankful I'm given the opportunity here to present, is the striking lack of communication, I think, from the science community towards media and public opinions, and how this is kind of really struggling for a lot of us. And I think we really have to improve on that. Future research, what we're going to do is continue to scale the testing at Baylor. We're working on fast and improved analysis methods and sequencing-based assays and rapid workflows to study diversity. So as I said, if you're interested in these topics, please feel free to reach out. And in the end, I just want to thank all my colleagues at HGSC, the group at Cornell from Chris Mason, Todd Trianglin's group at Rice University, NIH for funding, and I'm a member of COVID, which is a consortium where we come together, open science, to interact and exchange ideas. And with that, I thank you all, and happy to take any questions.
You mentioned seeing more diversity than you expected.
Yeah.
What does that mean to the layman?
Oh, sorry. So the main databases, right? So GCAT and others are just relying on whole genome assemblies, which means you're just having one continuous representation of the virus. However, what we identified are multiple non-consensus level variations that limit in the background, basically.
So you might say that you have there's actually multiple reference sequences?
Yeah, there are multiple reference sequences in each of the hosts. Yeah.
What does that say about the virus?
Well, I think so. I mean, the most likely cases that get infected by one virus type, which is the dominant virus type in your body, maybe. But there is a later point where it could even get co-infected with another SARS-CoV-2 derivative virus, and so you have like two slightly different viruses in your body. That could be one explanation. The other explanation would be pointing towards high mutation rates in the host body itself of the virus.
just because I know you're the person kind of for structural variants, did I catch you say that, you had less structural variants than you expected?
No, quite frankly, when I first discussed this with colleagues about if you would be able to identify structural variants in this virus, most of my colleagues meant that this is kind of pointless and viruses don't typically carry structural variants. So we were actually surprised and very curious and interested that we identified variations or structural variants that we believe are true, and especially at a higher frequency of 20% or higher for a couple of proteins. And these are like small deletions most of the time. That now I think also there are like 6 other papers that report a single deletion or something like that. But here we have a more comprehensive catalog of structural variants.
And again, for the layman, how might you, what does that mean to someone who may not understand everything around genomics?
So single nucleotide variants are just point mutations that are most prevalently studied. However, in other organisms besides viruses and humans and others, we see more and more the prevalence of structural variants that are just larger mutation types that have been so far understudied. That really showed a significant impact in evolution and other phenotypes and other human diseases. So my group was particularly interested to discover these types of structural variants and identify their roles. And I think this study was one of the first steps forward for that also in viruses and SARS-CoV-2.
I'm getting a question. Just what do you mean by structural variants for the layman?
Oh, I'm really sorry that I'm using expert jargon here. So as I said, in contrast, so we have single nucleotide polymorphisms or single nucleotide variants, which are just single character changes in the genome from a single to usually like 20 character changes in the genome. And then structural variants are really much of a more complex variant that are typically defined as 50 character changes or higher, that are really important for different evolutionary perspectives.
Can I maybe add, it means that basically a piece of the genome is basically either gone or it's been added.
Exactly. So the deletion means it would be gone, and then an insertion means that there's a novel sequence emerging in that virus.
Well, I think we've run out of time. Thank you so much for being here, Fritz.
Yeah, thank you very much for inviting me.
We have our next session starting in, I think, 12 minutes. I hope to see you guys there. Thank you again for Hewlett Packard Enterprise for sponsoring this session.